Yeah, so I'm going to talk about milestones in the scientific journey of Mary Bartlett Bunge. So this is a very, you've seen this image. So this is Mary at her electron microscope where she spent a lot of time, and that's Richard Bunge with her. And so I was a student of Mary's and Richard between 1993 and 1996. In fact, I really was Richard's student, but then he became ill, developed cancer, and so increasingly Mary became my mentor. And she aided in the completion of my thesis work after Richard passed away in 1996. She also was very encouraging of my interest in art. And then when I returned to the University of Miami, we held joint lab meetings between 1999 and 2017 when she started to retire. When I completed my Ph.D. the day of my defense, they gave me this pottery, which they had purchased on Martha's Vineyard, which I've treasured ever since. And so sort of the theme from this, if you see kind of the crack at the top and then the window in the middle, it was about fixing broken things. And that really is a major theme in what we do here in terms of spinal cord injury, brain injury, but also people's lives. And so I've always thought that this was a very wonderful gift that they gave to me. Mary went to the following institutions. She did her bachelor's degree at Simmons College. This summer she spent in Bell Harbor proved very instrumental for her love for biology. And then she had other plans, but she ended up getting an invitation from the University of Wisconsin, and she went there, did her master's and PhD work. Then both of them went to Columbia University. They had two sabbaticals that I'm aware of. One was at the end of the Columbia time at Harvard. Then they were at Washington University for 19 years, had a sabbatical in London, and then they came here to the University of Miami, where Mary was on faculty for 29 years. So Mary worked with Robert Schilling on her master's degree. So Robert Schilling was an internist, hematologist, who was very interested in the problem of pernicious anemia. And I was looking back and thinking about this picture, he looks like he's about 50. He was in his 30s, actually, when Mary was working with him. And he did many studies to try to work out the relationship of intrinsic factor as a binding component for vitamin B12. And Mary was involved essentially in biochemistry experiments. After that, something remarkable happened, which is that Mary somehow decided to work with Hans Ries for her PhD. Hans Ries was not a neurobiologist. He was an expert in genetics, and he was one of the new emerging experts in electron microscopy. He'd been at the Rockefeller Institute, and then he went to the University of Wisconsin. So on the right, you see one of his images scanning electron microscope of a chromosome. So anyway, Mary decided that this was a good person to work with. And there's a quote, "I saw the opportunity to combine the acquisition of scientific data with artistic expression." And I would say through her electron microscopy and also through many of the other beautiful panels of work that she created, and we all knew what it was like to meet her standard, which was very high, she really combined science and art very well. So this is sort of multiple EM images that have been pseudocolored, but to make the point that electron microscopy can be very beautiful as an art form, and it's capturing life processes instantaneously at a very high level of resolution. So I've always been fascinated with this particular paper. It was published in 1960. I started to look at this paper when I was a graduate student with Richard and Mary. And so this is the first demonstration of endogenous repair within the mammalian nervous system. And it's done by a guy who just finished his MD and a young woman who was finishing her PhD, together with Hans Ries, who was not a neurobiologist. So it's a fantastic combination. So what happened to make this paper possible was that Richard was working with an anesthesiologist, and they were doing a particular delivery of anesthetic at the base of the skull. And after they did it sometimes, he noticed the cats had a limp. And so they thought, well, there must be some type of injury there. And so they began to study these animals, and then they went into a very intensive study that had multiple time points, and they were only able to fix part of the spinal cord because the fixation techniques at that time weren't very good. So they were only able to fix the superficial layer of the spinal cord, but they were able to identify at 30 days after the injury that there was new myelin. And they knew that because earlier time points showed no myelin around these axons, then this very thin myelin, and then thicker myelin. And so this was really the first demonstration that the mammalian nervous system could repair its central myelin. So it was really seminal and coming from quite young people. So when I was working with Dr. Bunge, he also was very interested in these macrophages, and they actually came up in Elizabeth's presentation as well. And so he would call them Gitter cells, and we would look at these in human neuropathology. He would comment, they're there for years after the injury, what are they doing? And recently, there's been an interest in these as potentially neurotoxic cells. But I think that one of the things about Richard and Mary Bunge was they were very keen observers. And they made a number of observations that we're still sort of sorting through today, but they knew that what they were seeing was important. The other thing in the paper is that there's this beautiful diagram, and it is a conjecture of how central myelination works. And in fact, it conjectures that there's a relationship between oligodendrocytes and myelin. But they admitted in the paper, the connection between a glial cell body and a mature sheath has not been demonstrated. So this is what they thought was happening, but they hadn't been able to prove it yet. So at the end of the Ph.D. and the M.D., they moved to Manhattan. Must have been very exciting to move from Wisconsin to the big city at that time. So they were up near the George Washington Bridge, which is where Columbia Presbyterian is, in 1960. I can just imagine the excitement, the scientific excitement in New York City, the excitement about electron microscopy, and the new things that they could do. So one of the very first people they worked with at Columbia University was George Pappas. George Pappas was an anatomist, and he had mastered some really good techniques for fixation of tissue. And you can see the demonstration over there in this fine structure of dendrites in the superficial neocortical neuropil. So fixation was a big issue in electron microscopy at that time, and he was doing very well. And so they decided to work together. And in the next year, they published this paper. And so this paper is a seminal paper because it demonstrates that there's a connection between the oligodendrocyte and the myelin. People even thought maybe the axon made the myelin. So this was critical, and this is called the mesaxon, which is the beginning of the wrapping of the myelin. So this paper is very short, only has 2 figures, gets right to the point, and says that we believe that myelin is made by these oligodendroglia. So that was important work with Dr. Pappas. Now there was also somebody who was doing the other part, which is the part that Richard Bunge really loved, and that's Edith Peterson. So she was an expert in cell culture. She was particularly an expert in organotypic cell culture, which is the ability to keep organs and developing tissue alive in tissue culture and study it. And so Mary and Richard began to work with her, and they were taking developmental spinal cord tissue and they were studying it at different time points in tissue culture, and they were looking at the formation of synapses. So if you read in the paper, they would say that this is a relatively immature synapse, and then this is a more mature-looking synapse. So this is from a 14-day fetus that has been in culture for 69 hours. So they would tell us often about these long times that they would spend in the lab through the night, studying specimens, harvesting specimens at different time points so that they could find these events. So in the same set of experiments, there was a collaboration with Stanley Crane. So Stanley Crane turned out to be the expert on the neurophysiology of cultured tissue. And so in the same experiments with the same timing, he was studying the emergence of synaptic connections. He was providing stimuli to the tissue, and he was looking for the propagation, polysynaptic propagation, and he could find that at different time points. So my point here is that these were multidisciplinary collaborations, organotypic tissue culture, electron microscopy, neurophysiology, and trying to sort out the events that were pivotal in neurodevelopment. So then they moved to St. Louis, and this is one of Mary's first papers in St. Louis. And what you see here is time-lapse video of DRG axons coming out and contacting the cell body. So for many years, she was very interested in the mode and morphology of synaptic connections and how they became reinforced and stabilized. And you can imagine that if you look at this time lapse, we've seen this now many, many times with inhibitors of axon growth. So if you think about the work going back to Martin Schwab and Jerry Silver, they all showed this type of time lapse where there would be a growth cone that would connect with an inhibitor and it would collapse. So again, they were quite ahead of their time in terms of their techniques. Another thing that is in that same paper is a very careful measurement of the size of coated vesicles. And so this is a coated vesicle, and these vesicles they were observing, they were being exchanged between the presynaptic and postsynaptic part of these independent cells. They seem to be contributing to the formation of the synapses. And so just draw your attention to the 100 nanometer microvesicles that they were seeing here, and again point out that they were way ahead of their time. And so for a long time in electron microscopy, these vesicles were observed, didn't really know what they're doing. And so we're now very interested in vesicles that are about 100 nanometers, which are exosomes, and they are a little bit different than coated vesicles. But again, powers of observation ahead of their time, and just putting it out there as their data. They also, Richard in particular, when I worked with him, just such a great observer. And he would, every time you would go to a microscope with him, you would sort of see something new. So they use the tissue culture system now to figure out the critical events that were involved in the axon's relationship with glial cells, which then turned out to be pivotally important when we wanted to go to in vivo experiments with transplantation. So what's going on in this figure is that they're growing DRGs on a collagen substrate, and in some parts of the collagen, the axons have lifted away from the collagen. And so that would be the connective tissue part of this. And so this is a fascicle of many, many axons that would be sort of suspended in the cell culture. There's no Schwann cell activity in that fascicle. And so you can see here a fascicle that's forming with what we would sort of consider to be typical Schwann cell ensheathment. So they had a very clever idea. So when they had these floating fascicles, they would go back and lay on top of them a sheet of collagen matrix. When they did that, and they waited longer, they then saw that this turned into this. The Schwann cells migrated into the fascicle, and they began to perform ensheathment and myelination. So in this relatively simple experiment, they proved that contact with connective tissue matrix is required for normal interactions between Schwann cells and nerve fibers. And then here in the Miami project, we spent a lot of time, and many people in the room, including Martin Odega, worked on what that matrix should be to optimize the use of these cells for repair. Another thing that they did sort out is the critical nature of the contact between the Schwann cell and the axon. So this was another sort of smart experiment in tissue culture. And one of the themes of Mary's work was the basal lamina component, which is seen on the outer surface of Schwann cells. And this basal lamina appears to be critical for many functions associated with axon growth and regeneration. So in this experiment, they had these fascicles that were ensheathed by Schwann cells, and then they were able in the cultures to cut away the axons. They could go to the sides of the culture, just cut away the cell bodies, and then in a period of 3 days, the axons would degenerate. And when they did this, they progressively lost this extracellular matrix. And so when they had this observation, they went back in and they put new DRGs into the culture, and then these sort of disorganized processes of Schwann cells without extracellular matrix became normalized again. And so this is another example of just using the tissue culture system to answer fundamental questions. They then moved to Miami in 1989, and Mary had a really amazing list of graduate students and postdoctoral students, many of whom are here today. And this was quite an amazing run that she had with these 21 people. And if I forgot anybody, please forgive me. And I will touch upon the work of some people, and if I don't mention your work, please don't feel upset. There were only so many examples that I could give. So another thing about Mary that you may not know is that before she came to Miami, she'd had 45 publications. Many of these publications, she was the first author. This was her own work in the lab. After she came to the Miami Project, she had 113 publications. Richard Bunge passed away when Mary was 63. And after the age of 63, Mary had 98 publications. And I think that's a staggering achievement for a person. Many people would be thinking about retirement, but Mary was most productive after this age. So this is one of my favorite experiments that was done soon after Mary and Richard came, and this was work with Carlos Peno. And I think this was a particularly brilliant experiment looking ahead and in view of what we know now. So basically, this is collagen with DRG axons with Schwann cells. What they did in order to create an implant for the injured spinal cord that was inserted into the injury, they cut away the ganglia cells again, getting rid of the axons. That left them with the collagen and the Schwann cells. They then rolled this up and they inserted it into the spinal cord, and they had beautiful growth of fascicles of axons associated with Schwann cells. The reason this is so important is that we struggle very much with the survival of transplanted cells. And so we dissociate the cells from the surface using trypsin. But by not doing that, I think that the number of and quantity of cells that can be preserved alive is much greater. And so this is one of the first demonstrations of a biomaterial. Then something really important happened. There was a not so young scientist who was named Xiaoming Zhu, who came to be a postdoc with Mary. And this was a sort of coalescence of events. Veronique Guinard had come from EPFL in Switzerland, where she was working with Patrick Abisher. And Patrick Abisher was making these very interesting guidance channels. They had very interesting properties with respect to their porosity, and also with, I see these guys laughing here because everybody worked so much with these guidance channels, and maybe they weren't so great, but for the purposes of Xiaoming's work, they were. So the property was that you could take just cells with some matrix, put them in these guidance channels, and you would get a tissue cable. And that tissue cable, after insertion into a spinal cord injury area, would become vascularized and become part of the tissue of the animal. So these experiments by Xiaoming sort of set up a system that was then tested many, many times in different experiments. And so this became the bridging strategy. And so Mary had two main theme words. The first one was bridging, the other was combination strategies. And so in these studies with Schwann cell bridges, many things were tested. So it became sort of a system to build upon. And I've listed many of the types of things that some of the people in this room also assessed with this bridging system. So in terms of combinations, in 2004, Mary was working with Marie Philbin, and Damien Pierce was leading the study, and they decided to do a combination therapy, and they picked Schwann cells, Rolopram, and dibutyryl cyclic AMP. And when they put this combination together, they saw very abundant axonal sprouting, and they did see a significant increment in the behavioral recovery. So this appeared to be a validation of the idea that Schwann cells together with other therapies could increase the neurological recovery. And then Caitlin is here. I don't know where she is right now, but there she is. So I believe Caitlin made a major contribution to this work, because we know that when we release the cells from the tissue culture plates, they begin to die. And so Caitlin worked out very carefully by doing different stressors in the cell culture system, and also in vivo how these cells die. And so we now know that they die by anoikos, and they also die by apoptosis. And so in this particular paper with Mary, Caitlin utilized a number of inhibitors of apoptosis and showed that the survival in vitro and in vivo post-transplant was increased. This is still an unsolved problem for us in the use of Schwann cells. Then this will be the last experimental work I'm going to show. So this was by Ryan Williams. And so what Ryan was doing, among the many things that he did in his thesis, is that he was very interested in the interface between Schwann cell implants and the spinal cord. So this is the spinal cord over here. This is the Schwann cell bridge. And you can see that there's a very sort of conical configuration here, and then there's a much more fluid configuration. So he was introducing the Schwann cells when they were in a fluid state versus when they were in a gelled state. And when he did the fluid state, he observed that astrocyte processes were growing extensively into the bridge. And why this is so important is that this is taking us beyond something like a peripheral nerve. Much of what we were doing with these cables was very peripheral nerve-like, but now we're creating a novel tissue that doesn't actually exist in nature, because it's now comprised of these astrocyte processes. And this was of particular interest to me, because I had made exactly the same observation in my thesis work. And so Ryan kind of validated that. And so we think, it still remains to be completely established, that a bridge that includes astrocyte processes extensively throughout it should be a better bridge for the regeneration of certain types of central nervous system axons that require the presence of astrocytes. And then finally, this is the electron micrograph. You can see that this is a Schwann cell with its cleft nucleus, another Schwann cell that's myelinating. And in this bundle, and it was the role of endoneurial cells was brought up this morning, which are very interesting cells. Within this endoneurial bundle, we see the Schwann cell, axons, and the astrocyte process. And so this is still something for us moving forward to work out to a greater extent as to whether this type of hybridization between peripheral nervous system structures and central nervous system structures will improve a repair in the nervous system. Eventually, Mary was able to have this achievement of translating from all this large amount of preclinical work we did in multiple in vitro and animal systems to clinical Schwann cell transplantation. And Mary was very fond of the winged victory of Samothrace. And so I'll stop my presentation there. Thank you very much.